Good afternoon, everyone. I am Melissa Jones, and I'm the director for the Metabolic Disorders portfolio. I would like to introduce our patient as we move into the next session, Advancing Reasonably Likely Surrogate Endpoints. Now, our patient, Jackie Duda, happens to be a travel and health journalist and was originally diagnosed in 2017 with osteoporosis. Now, just 2 years later, she suffered a fracture while performing routine household activities. Both of these events really started her journey not only as a patient, but as an advocate with the Bone Health and Osteoporosis Foundation. So please join me in welcoming Jackie as she shares her story.
Thank you so much, and thank you to the FNIH for bringing me here today to talk to all of you, and to the Bone Health and Osteoporosis Foundation for making me an ambassador last year. My name is Jackie Duda. I live in New Market, Maryland, a little town just outside of Frederick, and I work as an advocate and a journalist. I advocate for sepsis, osteoporosis, and people with disabilities. I've been disabled since 2012. I also have Crohn's, POTS, hypermobile Ehlers-Danlos syndrome, and I added osteoporosis to all of that. As a journalist, I write about everything I go through, so I'll never run out of material. I met the FNIH last fall when they asked me to do that wonderful video, and I had the recent pleasure of interviewing Tanya and Dennis and Mary about the work that they did in this groundbreaking SABRE project. And that article just appeared on Saturday in the Lancet Journal of Rheumatology. So anyway, my story started actually back in 2015. I was diagnosed first with osteopenia. My doctor very wisely sent me for my first DEXA because I had so many chronic conditions. My mobility was terribly impacted by my EDS, so I used a mobility scooter occasionally. And I was taking prednisone and other steroids to treat my chronic pain and inflammation. The DEXA showed I had osteopenia, and it really didn't dawn on me that that was a bad thing. We don't have the education out there, at least like the patient education, that I feel we need so that we patients see alarm bells when we see that our bones are starting to degrade. It wasn't even 2 years later, my next DEXA revealed, yep, I have osteoporosis. Doctor says go to a specialist so that you can start treatment. Only problem was the specialist I went to wasn't interested in having a conversation. I mean, I'm a journalist. I'm very curious. I'm going to ask questions. And he wasn't answering my questions, especially about the side effects. So I went down the Google rabbit hole. I got really terrified of all the drugs, and I said, no way. And then my doctors all proved me right. In 2019, I suffered my first fracture. It was a thoracic fracture in my vertebra. 4 more joined it soon. Fractures seemed to like company. And then my life got totally interrupted when I almost died in 2021 of septic shock. I had undiagnosed diverticulitis that led to a colon perforation. I had to be flown to Baltimore to shock trauma. I was near death. So that was sort of a turning point. And I had to undergo very intensive rehabilitation, physical rehabilitation, because sepsis rendered me unable to walk. It also destroyed my muscles. If you see my Instagram, I posted a picture on there last night of me in a shorts, sort of a bathing suit. I had no muscles. I was literally skin hanging off bone. So I had to do all of this intensive exercise. And in 2022, I was well enough to join the YMCA again. And I started working out there. So that was my major flex. Hey, I'm doing great. My bones are going to do great. Nope. So my bones betrayed me once again. And my DEXA in 2023 showed that, yeah, you did a lot of exercise. Yes, you're a lot more active now. You eat well, but your bones are still declining, and they were worse. They were way worse. T-scores were -4. And then I fell playing with my golden retriever, and I fractured my left wrist. And that was it. I thought, okay, I can't run away from the medications anymore. I got to find a doctor. But I got to find a doctor who wants to talk to me and will talk to me about the drugs so that I feel reassured. And sure enough, I did. I found a wonderful endocrinologist at Johns Hopkins. He likes to talk as much as I do, so I have no problem with that. And then he even explained to me the clinical trials, how they were conducted, you know, why those rare side effects occurred. And then I felt completely confident, and I said, you know what, doc, okay, I'm going to listen to you. And I started to use it was a bone builder drug because I was way far gone past bisphosphonates at that point. And I used that bone builder for 2 years, which took me up to last year. Well, I did have a DEXA also in 2024 because once I started it, my endocrinologist wanted to do the DEXAs every year. 2024, I did show some improvement. 2025, I showed major improvement in my bones. My hip even went back into osteopenia range, which was incredible. So I swim and work out. High-intensity water aerobics is the key for me because of pain, and water helps my pain. And I do get weight-bearing because we use weights and other equipment in the water. And last year I was able to join my daughter Elise and do the turkey trot on Thanksgiving morning in Frederick. I walked it, but my time was under an hour, so it wasn't that bad. But I did, you know, my journey's come all the way down the timeline to there. And then the ultimate proof to me that the medication worked, I was at the beach last year with my kids. A wave hit me. I was right in that danger zone. A wave hit me, and I went down straight out on both of my wrists and my knees in hard sand. I didn't break a thing. When 2 years prior, I fell and broke my wrist just by my dog bumping into me. So that I'm convinced the drugs work. My mission now is to inform other patients through my journalism, my social media, and my podcast, which has 81 subscribers. It's getting very popular. But I do invite scientists. I interview sepsis survivors, people with chronic conditions, people who've lost someone to sepsis. And I also have had a vaccinologist on there. I have had physicians on the podcast with me. I'm going to interview a nurse tomorrow about blood culture contamination because I'm trying to be the conduit to get all this wonderful information and work and research that you all do out there to a general audience of non-sciencey people like me, and to inform patients that the medications work, the osteoporosis medications are safe. Don't go down that Google rabbit hole. And science works, and scientists are trying to help us, and there's nothing to fear. So thank you very much. I appreciate being here, and thank you for listening to my talk.
Thank you so much. And now I would like to introduce Jennifer Hamilton, the Vice President of Precision Medicine at Regeneron.
Thank you, Melissa. I'd like to thank the FNIH and the Biomarker Consortium for giving me the opportunity to speak and to moderate this panel today. I don't know, I think that it's been a very inspiring and energizing event for me personally, and so I think I'm excited about how this conversation is going to go. So the FNIH Biomarker Consortium is a key example of community team science. So I'm going to play on what Dr. Gerberding said yesterday about team science. This isn't just a team. This isn't just an individual team. This is a whole community of different fields of study pulling together with a common mission. And with the Biomarker Consortium, we're leveraging pre-competitive projects to advance the use of biomarkers as endpoints. That's one of our key goals. I thought Dr. Woodcock said it best yesterday when she highlighted that the field of biomarkers is hard. I felt seen. I don't know about the rest of you, but I felt seen. I often think that it's not rocket science, it's harder. So we're here celebrating 20 years of pre-competitive advancements in the use of biomarkers to improve the practice of medicine. Over the last couple of days, we have heard about the advance of biomarkers across indications. It's not just oncology anymore, even as personalized as the inspirational stories we heard from Dr. David Feigelboom this morning. We've heard from colleagues looking to utilize blood markers that may be informative enough to alleviate the need for invasive procedures, minimizing patient burden, and are better for longitudinal measures in clinical development. And we've heard about how AI and machine learning are advancing the field of pathology, imaging, and beyond, and all of the new challenges that we have in advancing such markers to the clinic. It's no coincidence that we saved this session for last. That's just for you, Amanda. So why have we saved it for last? Well, we've advanced the field significantly together over the last 20 years, but there's so many more opportunities to apply biomarkers to accelerate the speed in how we are getting new, safe, and effective treatments to patients in need. The application of biomarkers as endpoints has the potential to shorten the duration of clinical development by reducing the need for longer, larger studies with clinical outcomes. So now we have accelerated approval, which we're going to talk about in a moment. And think about the patient's perspective that we just heard from Jackie and her battle with osteoporosis. It used to be necessary to conduct these large, long clinical trials to demonstrate the reduction in fractures with statistical rigor, right? So the qualification of DEXA as a surrogate for fracture risk will reduce the size and duration of future studies. This is going to be a game changer for drug development in osteoporosis, and we'll be hearing more about that from our first speaker, Dr. Dennis Black. Before we dive into those presentations, I think it's important that we start out on a common ground with biomarker endpoint terminology, which is surprisingly inconsistent, confusing, and clear as mud. And so part of our goal for today is we're going to talk about the different terminology that we use for biomarkers as endpoints. We're going to review the development and regulatory acceptance pathways of biomarkers. And speaking from the drug development industry, there's a lot of clarity that's needed from sponsors about what is the weight of evidence required for biomarker endpoint strategies, and what are the different mechanisms that we have to advance these biomarkers for use as endpoints without having to do what was it, Dennis, 12-year qualification program? Yeah, that doesn't work on a pharmaceutical timeline. So the first thing we're going to talk about is what is accelerated approval. Now, this is an FDA-centric pathway. We do have our colleague Thorsten Vetter, who's going to be joining us online from the EMA, but most of the terminology that I will be talking about is very FDA-centric. So accelerated approval is intended to facilitate and expedite the development and review of new drugs. There's an additional condition to address an unmet medical need in the treatment of a serious or life-threatening condition, and then there's a very specific definition of a serious or life-threatening condition, which I suspect is also heavily debated amongst different people. I'm not going to read it to you. You can look at it later. And the idea is that we fill an unmet medical need based on a surrogate endpoint or an intermediate clinical endpoint that is reasonably likely to predict an effect on irreversible morbidity and mortality or other clinical benefit. So there's these three terms we've just thrown out: surrogate endpoint, intermediate clinical endpoint, and reasonably likely. So, validated surrogate endpoints. Again, if you look at the guidances and the definitions of these biomarkers, a validated surrogate endpoint has completed the FDA biomarker qualification program, and the additional confirmatory trials are not needed. And an example that we'll hear about today is the bone mineral density DEXA scan for osteoporosis. Reasonably likely surrogate endpoints support the strong mechanistic and/or epidemiological rationale such that an effect on the surrogate endpoint is expected to be correlated with an endpoint intended to assess clinical benefit in clinical trials. That's clear to everybody, right? Okay. Without sufficient clinical data to show that it's a validated surrogate endpoint. So this is an opportunity for us to not go through a 12-year qualification progress for a fully validated surrogate, but there's a lot to unwrap here about what does this actually mean. And if a reasonably likely surrogate endpoint is used for accelerated approval, the sponsors will be required to do confirmatory trials. Now, I think intermediate clinical endpoints, I personally struggle with trying to figure out what the difference between reasonably likely and intermediate clinical is, so that's going to be some of the questions for the panel today. It's a measurement of therapeutic effect that can be measured earlier than an effect on irreversible morbidity or mortality. Demonstrates clinical benefit on a less serious or earlier symptoms of a serious disease, but the benefit observed is anticipated to predict a favorable disease outcome. Again, this is another pathway for us to use endpoints, and oncology has been doing this for a number of years, and we'll be hearing from Nicole on this in a few moments. So with that, everybody has the same understanding. We're all using the same terminology. My personal pet peeve in the field is the use of the term surrogate being thrown around too loosely, so hopefully none of you will do that when you walk out of this room today. But with that, I'd like to turn this over to Dennis Black from UCSF, who is going to tell us about the DEXA project for osteoporosis.
Thanks, Jennifer. And I just want to thank the FNIH and the Biomarkers Consortium, as you'll hear in my presentation. They played a key role, and we've all, stuck together for this long time. And also to tell everyone else, I think if someone is going to go through this process again, I think the fact that we've already gone through it will be helpful in accelerating it in the future. So don't be too intimidated. So I'm going to talk about the SABRE project, Study to Advance BMD as a Regulatory Endpoint. I think I'm actually... it was formerly the FNIH Bone Quality Study, so you may have heard of it in that terminology. So again, as everybody's been talking about, this is team science. I've never done anything like this with an incredible group of a broad-based coalition of stakeholders that have worked together on and off for all these years, and that includes society and foundation partners, the ASBMR, and of course the FNIH, government partners, NIAMS, NIH, and FDA, and I particularly want to call out NIAMS and Gail Lester. I don't know if Gail's here. We invited her. Gail was at NIAMS and is now retired, but she and Mary Buckstein, my colleague at Harvard, and I remember in 2013 sitting down in a cocktail bar in Baltimore, the three of us, and Gail saying she had worked with FNIH on osteoarthritis and thought it would be a really good fit for us to try to do this project. So she's really the mother of this project, and the credit should go fully to her. In any case, we also have academic groups. There's our group at UCSF, Mary at Harvard. There's a group at University of Sheffield, and then we've had advisors from Mayo and University of Pittsburgh. So we have a broad-based group. And then the industry partners, I want to particularly credit them because they supplied two things in this study. They supplied the raw data, and that was when I'll talk about that in a minute, but getting the individual patient data was a real challenge, and we worked with a lot of companies, as you'll see in a minute, and you can see the names here. And also they supplied a lot of the funding that got this project going. So really it was a really good group, and this would not have been possible without FNIH. I also want to just mention my study team at UCSF. All these people on the list here have been with the project for now over 12 years. So the statisticians who've done miracles with putting together this final package for the FNIH, for the FDA, and also doing all the data analysis. Okay, let's go here. So what background here? Okay, I think we've heard about this already. There's a strong need for new osteoporosis therapies. We have current treatments that are effective, and they're quite effective, but use is declining, as you can see from this figure here. And for example, there's a 60% decline in osteoporosis med use between 2007 and 2018. And adherence is also poor. And a lot of this is due to publicity about some very rare side effects. They're extremely rare, and we've done some benefit risk calculations, and it's overwhelmingly positive. But despite that, people are afraid. And so these drugs have just become difficult to really have people use them and continue to get the efficacy that they would get. On the other hand, development of new treatments is challenging. I think our field is not unique in having an ethical need for active comparators and or low-risk subjects. You can't treat people with placebo when there are effective treatments. And fractures have been the primary endpoint required by FDA and other regulatory agencies for approval. And this has led to very large trials, up to 15,000 patients for one, that went on for 5 years. And they're very expensive, and it's too expensive, and it's no longer feasible to develop new therapies. And currently, there are no alternatives to fracture endpoints. I should say currently, now there are, but there were not until just last month. And there were no osteoporosis medications in late-stage clinical development. So the project, as you've heard, began about 13 years ago now to develop a surrogate biomarker, a validated surrogate biomarker that I now know how to name what we're doing. And again, public-private partnership, as you've heard. And we decided in the beginning to go on this hard route of collecting individual patient data from randomized trials. And there's some value in the individual patient data in being able to pool it across all these trials. That's good. And because of that, we could standardize our endpoint, which was fractures, and standardize the biomarker, which is bone density. There are several different machines that measure bone density. And so we could do that because we had the individual patient data. We could also respond to FDA. For example, they asked for subgroup analyses in women at high and low risk. So we could do all those things with the individual patient data. So it is valuable. And the database is also a pretty valuable resource. I mean, we have, as you'll see in a minute, about 170,000 patients. And we've written about 15 papers on comparing efficacy in different kinds of groups, et cetera. So again, special call out to the FNIH for their role in facilitating this project. So what is our surrogate? Our surrogate is actually 24-month % change in total hip BMD active minus placebo. You can see here a picture of a DEXA machine. DEXA is a really nice biomarker because it's been around since about 1990. It's used clinically. It hasn't dramatically changed, unlike many biomarkers. So it's very similar. So all these trials that have been done since that time, pretty much we could pool all the endpoints. And so the biomarker is specifically, I guess this is not, I think, no, it's not the right slide, but in any case, is the 4.7%, which is the difference between the active and placebo group at 2 years. So that's the biomarker's value for this particular trial, which is a trial of zoledronic acid. Okay, so in terms of our data compilation, you can see that we worked out with a lot of different drugs, all the approved drugs we have the individual patient data for, as well as other drugs for which approval was not sought. For example, some of the SERMs, Donacatab, when they got all done, they decided the company was not going to submit for approval. So we collected data from 53 studies, a total of about 176,000 patients. So I think this is a bar that probably the next person or the next group that intends to get this approved may not have to go quite this far. But that's what we chose to do. It would be easier now because there's better publicly available data from clinical trials mandated by journals and by various sources. So the primary analysis that we did is a meta-regression with 2-year total hip BMD versus % change in fracture reductions. And so what you see on this graph, it's a little hard. I don't think I have a pointer here, but just to kind of tell you, on the horizontal axis, you see the biomarker, the percent change in total hip BMD, active minus placebo at 24 months. On the vertical axis is the hazard ratio, which is the reduction in fracture risk in that trial. Each bubble represents a trial, larger bubbles, larger trials in general, and then the colors represent the different classes of medications. And basically what we do in the analysis is you fit a line through these. It's not quite, it looks like a straight line. It's pretty close to a straight line, not exactly a straight line. In any case, and then calculate the correlation. And so in this particular example, which is one of our fracture endpoints, which is all clinical fractures, that includes hip fractures, wrist fractures, clinical vertebral fractures, and BMD changed, the correlation was 0.71, which is quite good, very strongly significant. A value of 0.65 and above is considered to be adequate for surrogacy. So the FDA agreed with our conclusion in our final package that we sent to them, and of course approved this. So how would this be used in the future? So thinking now, we've kind of turned our attention to thinking about what's going to happen now, and there are a number of companies that are kind of coming into this space based on this qualification. So the idea is we establish a threshold for change in BMD for which the drug would have a 95% chance of reducing fracture. And this is a technique called the surrogate threshold effect, and it was developed in about 2016 by Ciani. I think that reference may have gotten left off the slide, but in any case, you can see here this illustrates the calculation of the STE, and in this case it's 2.04%. So what does that mean for a future drug developer? If a future trial of a new drug showed a percent difference in the active and placebo groups at 2 years of greater than 2.04%, then that drug would almost certainly reduce risk of all clinical fractures and could be approved. And so that's the kind of the basis. So we created thresholds, and this 2.04 for this all clinical fracture endpoint. There's thresholds for hip fracture, vertebral fracture as well, and I think it's up to the FDA to decide which threshold they're going to use. But this is not a big threshold because most of the drugs that are approved currently did show changes larger than 2% in their trials. So we think that new drugs, particularly anabolic medications, which show larger changes, will qualify based on this criterion. Turning a little bit to the FDA, I think everyone in this group probably knows what's involved at the biomarker qualification program in general. And we were the first group to go through this for a surrogate endpoint. But the first step is a letter of intent, and then the qualification plan, which is, for us, was basically a very large analysis plan. We didn't collect any new data. We used this existing data that we had compiled. In other cases, other kinds of endpoints or biomarkers, you might actually have to do a study, but we didn't do that. We just did a long analysis. And then the last step is the full qualification package, which is the briefing book. So we initiated the process with FDA in 2017. In 2018, when the 21st Century Cures Act was approved, there was a slightly different qualification program that was initiated. So we kind of had to convert to that in 2018. We went back and forth a couple times with our qualification plan with FDA, and finally it was approved in 2022. The full qualification plan was a long document because we had all the analysis we did. We had the FDA also wanted the computer code that we used to generate all the analysis. So it was a long document, about 400 pages in the end. But and anyway, that went in and finally approved in August of 2023 with some revisions, and so we did those revisions, resubmitted in January of 2024. And we've been waiting for a while, but I know that the process is very complex. There many committees within FDA that have to review it, and then it has to come back again to higher level groups. So it did take a while. I think, again, the next one through should be quicker. But we got a nice Christmas present from the FDA on December 19th last year, and that was that we were qualified, that we received the full qualification. So it was very exciting for us, and I know it's been mentioned several times at this meeting as well. So, and you can see the FNIH, again, was very helpful, working with Tanya in particular, Tanya Camphouse, to kind of get this out into the world. So it was really, we had a kind of a campaign with the press release from FDA, which you can see the link from that they did, and FNIH did a press release, MGH did a press release from Harvard, and this really got picked up. There were a lot of news stories. There have now been several editorials that have come out in journals, and I know there's going to be one in the Journal of Bone and Mineral Research, and we're just finishing up one for JAMA. There was one last week in Lancet Diabetes and Endocrinology. So this has gotten a lot of publicity, and so I think this will generate a lot of interest in other groups in trying to do this. And this is just kind of a summary, 12 years, but fairly modest funding. I mean, the $4 million was, retained all our efforts, a staff of about 15 people on and off for 12 years. So I think it's really doable, and I hope that people are not intimidated by some of the numbers that I've shown. Again, just some of the other publicity and different articles that have come out in the last few weeks. Just to end, to talk about BMD as a surrogate, we are confident it will transform approval for new osteoporosis treatments. To give you an idea, again, these trials that had been done most recently were very large. All the trials from the beginning, even for alendronate, which is the first one, was like 7,000 patients. So they typically would have as much as 10 to 15,000 patients, and the trial length would go on for 3 to 5 years. With the future BMD clinical endpoint, we did a number of validation studies and recommended to FDA that the study could be as small as 500 patients. That could be 250 per treatment, so modest trial that could be done, and only 2 years of follow-up is necessary for this. So this is going to make it much more feasible, and I think really show the value of using a surrogate endpoint. So what will we get out of this? Of course, quicker, cheaper new drug development. There are, again, already at least 4 or 5 companies that are coming into this space and talking with FDA and talking with us about how they could implement this trial. And ultimately, the most important thing here is we want to generate more choices for patients like Jackie and others and clinicians, so that with more choices and perhaps different ways of administering drugs, different periods of duration that the drugs need to be taken, we hope we can really make an impact on the crisis in care in osteoporosis. And again, it's for a surrogate endpoint. I think you know that. And again, I think a model for future surrogate endpoints in other areas. And I know I've heard about a lot of them, and you'll hear about more in this session. And we are definitely open. You can contact our group at UCSF to help anybody who might need some advice. And I think you can find all of our documents are on the FDA website, but we'd be happy to help anyone who might want help with this. Okay, so I guess that's it, and thank you very much, and I'm happy to answer any questions in the panel.
Thank you. So my name is Michelle Long, and I work for Novo Nordisk, and I'll be talking to you about hopefully a new opportunity and maybe a future success story, let's hope. We talked a little bit about MASH, but MASH is a serious chronic liver disease. It's basically the manifestation of the metabolic syndrome in the liver. It is a slowly progressing disease, but ultimately patients with MASH can go on to get end-stage liver disease, cirrhosis, cancer, decompensation. A patient advocate once told me that the diagnosis of cirrhosis is emotional death. it is akin to a cancer diagnosis, even if it doesn't come along with cancer, but unfortunately it very often does. And this is also because the disease is silent. People feel fine, and they don't know that this has been manifesting in their liver for, their entire life. Until very recently, there haven't been treatments for it. So we are working within the accelerated approval and conditioning marketing authorization because of this. This is considered a serious disease that has a huge unmet need. And so for drug approval in this area, now these are patients who do not yet have cirrhosis, we run a clinical trial, and we enroll it based on identifying patients on biopsy. So we do a liver biopsy to characterize the disease, and then we look for changes in those parameters as a reasonably likely surrogate endpoint that is thought to predict clinical benefit if we can show improvements on the features of MASH and fibrosis in the liver. And then the trial continues to clinical benefit. So for the two drugs that are approved via the accelerated approval pathway and also conditioning marketing authorization in Europe, the trials are still ongoing to clinical outcomes. So there are no validated surrogate endpoints in this field, and really our patients very much rely on having access to these drugs as quickly as possible via these pathways such as accelerated approval, which has been really transformed and really allowed drug development to occur in this space when you're talking about a disease that progresses over 10, 15, 20 years. So the current state of affairs, really where we are with the histology-based endpoints that I mentioned to you, were highly innovative. I mean, it was a meeting of the minds, scientific community, sponsors, FDA, now back in around 2014, to really come together about what do we think would be reasonable to show in a clinical trial that might be predictive of benefit. And in these days, the biomarkers were very immature. So it was the best that we could come up with at the time, and it made a lot of sense. And it certainly has allowed drug trials to be accelerated and come in. And there was a lot of activity in the field, many trials coming around 2015 in that frame, from there to around 2020, lots of activity. But in this time period, we also learned some hard lessons. We touched on it a little bit yesterday, some of the challenges with the histology. So when you're identifying patients on histology, there can be disagreements with the pathologists, and really identifying some of these, what we consider key features of MASH, can be very hard to identify. And this, unfortunately, also results in very high screen failure rates for trials. We find that the screen failure rates, mostly on histology, can range up to 75, 85%. So when you're doing a trial, you're enrolling patients on biopsy, you do another biopsy at 52 weeks, and then you do a biopsy at the end of the study to see if they've progressed to cirrhosis or developed other clinical endpoints. And when you do the math if you're looking at an outcome study, about 1,200 patients, you're doing biopsies on you're doing approximately 10,000 biopsies. Some of those patients will get 3 biopsies during the course of treatment. This is only for, if it's 2-to-1 randomization, we're talking 800 patients that, could potentially be actually getting active drug in this program. So it's a heavy burden on participants, it's a heavy burden on society, and it's very expensive for sponsors as well. And we are definitely seeing, it was a little, gave me a shiver when Dr. Black showed that there was no osteoporosis trials in late stage development. And I really hope that's not the tale we're telling in a few years for MASH, but it very well could be, because I think we are seeing a huge gap in completed phase 2 trials being able to go into phase 3 because of the enormous cost that it comes with running these highly complex trials. And even to this day, we still do not have data showing that what we think is a reasonable endpoint in MASH on histology actually predicts clinical benefit. We don't know that this one-stage improvement in fibrosis, as defined by a pathologist, actually shows clinical benefit. It still is considered reasonable, of course, but we haven't seen that. And all along this time, the field has changed. And liver biopsy is now not the standard in clinical practice. And biomarkers have emerged, particularly imaging-based biomarkers and also some blood-based biomarkers as well. And so we see that liver biopsy is very infrequently used in clinical practice. We see with the new drugs that have been approved for therapy that guidelines have come out as well. Even the label for how these drugs are to be prescribed is that they have to have disease consistent with a certain fibrosis stage. So acknowledging that most people are not going to actually get that biopsy and know the fibrosis stage based on biopsy. So along this line, there have been several large epidemiologic studies that have come out as well, showing mounting evidence that there's a link between non-invasive tests and outcomes. These are, so this is a little bit the state of affairs where we are. So this is why we wanted to bring this discussion today as well, about thinking about what are the pathways to innovate here and develop new reasonably likely surrogate endpoints in MASH. You can propose new ways of advancing biomarkers in several ways. It can be a sponsor going to a particular, in the process of a particular compound, having discussions and meetings with the health authorities. It could be through scientific community consensus. That's how we got here to begin with the histology endpoints. Or it can be through the biomarker qualification program, and that will be primarily the focus here today. So we are very excited to say that over the last 6 months, there have now been 2 letters of intent accepted through the biomarker qualification program by the FDA to look at 2 separate imaging-based biomarkers as potential reasonably likely surrogate endpoints to be used in MASH clinical trials. But the hard reality is, it's not as Janet said yesterday, small crappy studies, but these are large studies that are out there, but the endpoints are not all the same, and the time points for where the biomarkers are measured out in the world are not the same. And there's non-interventional studies. So, there are challenges to looking at these natural history cohorts that we need to face and understand, and really, I think, we talked a lot about the black box, but this is a little bit of a black box, not related to AI, but still a black box of what does it mean to qualify something as reasonable? I think I'm a reasonable person, but my 14-year-old does not. So, we disagree. And that's natural. But I think what we're trying to understand is, how can we agree on something that might be reasonable and help spur future trials and drug development, while we also build the case and lay the foundation for validation. So that's a little bit where we're going here. So we have now an FNIH reasonably likely surrogate endpoint working group for MASH that has come from out of the metabolic disease area. And we're building actually on several projects in MASH that have been conducted with the NIH, transforming how we do trials in MASH. So this was the NIMBLE project that is still ongoing, but looking really at can we use these non-invasive tests for diagnostic enrichment. And then second, a project called MASH Track, which I also co-lead with my academic collaborator, where we're looking at prognostic context of use. So can we look at these non-invasive tests or NITs, and are they prognostic of outcomes? And we've just recently actually described this work that we're doing, and it was just accepted in Nature Medicine. So look out for that if you want to hear some more, but I'll also be happy to answer questions during the discussion. Thank you.
Hi, my name is Nicole Gormley. I'm a hematologist and director of the Division of Heme Malignancies 2 at the FDA. And so a lot of my examples will come from the heme malignancy space. I'm also the Associate Director for Oncology Endpoint Development within the Oncology Center of Excellence. Congratulations to FNIH for 20 years. It's quite monumental. And thank you for the opportunity to share a little bit about what we do. So I just want to level set as well a little bit at the start of this talk and really sort of underscore that there are really different uses for biomarkers. We talk about clinical uses and regulatory uses, and the information needed to support and justify use in each of these contexts is different. And I'll just touch basically on some of the regulatory uses, including patient stratification, selection, enrichment in trials, risk-based treatment assignments, making treatment decisions based on various biomarkers, and then as a potential endpoint as well. And there's really increasing risk associated with those biomarkers in each of those settings as listed on the slide. So as we've heard today, there's a lot of different terminology. And one of the things I'd like to just, highlight is that for regular approval, it's really, approval is based on demonstration of clinical benefit or an established surrogate. Whereas accelerated approval can be based on a surrogate endpoint that's reasonably likely to predict clinical benefit or an intermediate clinical endpoint, which should also, be reasonably likely to predict clinical benefit. And so when we talk about clinical benefit, we're talking about the way a patient feels, functions, survives. A surrogate endpoint is one, as we've heard, that's been fully validated and is not itself a measure of clinical benefit, but predicts clinical benefit. Surrogate endpoint, reasonably likely, or an intermediate clinical endpoint, those two are both used for accelerated approval. And with intermediate clinical endpoints, it's a therapeutic effect that can be measured earlier than morbidity or mortality, but again, still must be reasonably likely to predict clinical benefit. So I'm going to start out. Most oncology endpoints are not surrogates. And we have, in our accelerated approval, most of these are actually intermediate clinical endpoints. And we've used overall response rate, progression-free survival, event-free survival, and we deem them as intermediate clinical endpoints, as there's not robust data to support that they are surrogates in any shape or form. And oftentimes, there may even be data to suggest that they're not surrogates. One example that I'm not sharing today, but frequently talk about in multiple myeloma, is where we've had improvements in randomized trials with improvement in response rate, improvements in progression-free survival, and minimal residual disease, but still worsening of overall survival. So this really, MRD, is oftentimes, it's not a surrogate. So most oncology endpoints are not surrogates. In order for a biomarker to be a true surrogate, it must be in the causal pathway of between the treatment of the disease and the true clinical endpoint. And in this figure, the biomarker is within the causal pathway, and the intervention directly impacts the clinical endpoint of interest and the biomarker through that. The classic example would be, CD4 count in HIV, and one could even argue, BCR-ABL PCR in CML really sort of has that same fundamental relationship. But this, again, is not very common in oncology. More typically in oncology, there may be multiple pathways through which the disease has an impact on the true clinical endpoint of interest, and not all of them may be measured by the biomarker of interest. Additionally, the intervention may have differential impact on the different disease pathways, and it may even have impacts on the true clinical endpoint separate and apart and outside the disease causeway. And so this is why within the oncology space, it's actually very rare that we have surrogates. So oncology endpoints, generally we don't have surrogates, but that's why we rely a lot on these intermediate clinical endpoints. So historically, this was discussed a little bit yesterday, the Prentice criteria has been set forth as a set of criteria that can be used to validate a surrogate. And really, it's a set of statistical assumptions really that sort of state that the treatment effect on the surrogate should capture the full treatment effect on the clinical endpoint. The Prentice criteria really aren't thought to be as attainable or realistic, so a lot of times we've used meta-analyses and other such statistical methods to really assess the relationship between a biomarker and long-term clinical outcomes of interest. So we've heard a lot about this, but I just wanted to share a little bit again from the oncology perspective. There are multiple pathways through which a biomarker may be accepted by the FDA. We've heard quite a bit about the FDA biomarker qualification program, and this really the purpose is that within a stated context of use, the biomarker can be relied upon to have specific interpretation and application in drug development and regulatory review. There's also the opportunity for collaborative group interactions with the agency, and then as well within a specific drug development program. And I'm going to talk a little bit more about the last two. So one example of a collaborative group interaction with the agency is the use of minimal residual disease in multiple myeloma. I'm going to talk about that quite a bit throughout the rest of the talk. But then within a specific drug development program, one example is the approval of blinatumomab. And this was approved for the treatment of B-cell ALL in first or second remission with MRD greater than or equal to 0.1%. And really, there was submission of patient-level data for this approval for two aspects of this submission. And the first was that to identify a population at greater risk of relapse with a certain MRD level. And then the second was what reduction in MRD level would best predict long-term outcomes. And so the MRD data, patient-level data, was submitted to the FDA to support this application. And I think one distinction that really should be emphasized here is that with that approach, this really is only useful within that specific drug development program, whereas the collaborative group interactions, which I'll describe later, can be used more broadly within that disease space. So I'm going to shift now and just really talk about the example of the minimal residual disease and multiple myeloma. And it's really important here. One of the things we talk about is actually the need for development of novel endpoints. And I think multiple myeloma, there was really a great need. In the past 20 years, we've had approximately 20 approvals of new therapeutics, which have significantly changed the clinical outcome of those patients with overall survival. In this disease space, we're having, even in the relapse refractory setting and very relapse refractory setting, exquisitely high response rates that are sort of matching or approximating the response rates seen with combination therapy in earlier lines of setting. And in the newly diagnosed patient population, we're having response rates that are greater than 90%. And so a situation arises in which it's really not feasible to conduct clinical trials using overall survival, and it's very challenging even to devise trials based on response rate that are showing a benefit over other previous therapies when that baseline is at 90% without having very large clinical trials. So there was really a lot of interest in development of minimal residual disease within the disease space of multiple myeloma. So to that end, talking about team science, there were two groups really that collated the data that was available. This was the I-Squared team and the University of Miami. They worked together with partnerships with industry, other academic centers, and collated the data and submitted this collation of data to the FDA for assessment and evaluation of MRD as a potential endpoint to support accelerated approval in multiple myeloma. And basically, just to dive into these terms a little bit more, they assessed with the submission of the data the individual level association. Again, this is all based on patient level data, raw level data. And they assessed the individual level association, which is the strength of the association between MRD and the true clinical endpoint, which was progression-free survival and overall survival in this instance. And then also the trial level association, which is the strength of the association between the treatment effect on the surrogate and the treatment effect on the true clinical endpoint. Again, not really necessarily looking to validate this as a surrogate. In order to validate this for a surrogate endpoint, which again would have supported traditional approval, there would have need to have been, trial level association supported. So the results, they submitted the data to the FDA. There were ultimately 25 comparisons that were able to be conducted and data comprising over 11,000 patients. And this was assessed in the newly diagnosed transplant eligible population, newly diagnosed transplant ineligible, and then the relapse refractory patient population. And ultimately, this is sort of just a high-level overview of the results. On an individual patient level, there was very strong association for both progression-free survival and overall survival, and moderate to strong associations for the newly diagnosed transplant-ineligible population as well. Weak or moderate or no associations were seen for the other categories assessed, including overall survival. So this was presented at an advisory committee meeting at in April 2024, and it was generally, it was resounding support for the use of MRD to support accelerated approval in multiple myeloma. The committee voted 12 yes in support and 0 no. And then most recently, just this past month, draft guidance was released from the FDA on the use of minimal residual disease in multiple myeloma as an endpoint to support accelerated approval. And the guidance really discusses various trial design considerations, statistical design considerations, and assay considerations for use of MRD in this disease setting. And then additionally discuss the use of CR as an endpoint to support accelerated approval in multiple myeloma. So in summary, novel early endpoints really can expedite drug development and have the potential to get novel therapies to patients faster. And I think the need for novel endpoints, especially as demonstrated in the multiple myeloma example, is really critical to consider. We rely on patient-level data in oncology, and one take-home point, most endpoints that we use within the oncology field we consider intermediate clinical endpoints and not surrogate endpoints, and not surrogate endpoints reasonably likely to predict clinical benefit, but rather intermediate clinical endpoints. And then there are risks associated with the use of early endpoints, and so these must be balanced with the potential advantages, and really our decisions to rely on such endpoints should be data-driven. So thank you very much, and I look forward to the discussion later.
I would like to thank all the speakers for their absolutely wonderful talks, and let everyone know that we're going to adjourn for a brief break from 2 to 2:30.
So we had some great examples of success stories in validating surrogates, and we've heard about how intermediates clinical endpoints are used in oncology. And we've heard about, kind of the next horizon of markers that will probably not be, that may be fully validated surrogates, or they may go down other reasonably likely surrogate endpoint paths. So it's really an honor to be moderating this panel today. We do have a few people who are part of this panel that did not speak earlier. So maybe we'll start with Han. We'll just go down the line. Han Cho, can you introduce yourself briefly?
Yeah, thanks. My name is Han J. Cho. I'm the Chief Medical Officer for the Multiple Myeloma Research Foundation.
Hi, everyone. Danielle Graham. I lead the Biomarkers and Systems Biology group at Biogen.
Good afternoon, everyone. I'm Jeffrey Siegel. I'm Office Director for the Office of Drug Evaluation Sciences and the Center for Drugs at the FDA. Our office is responsible for the qualification program for clinical outcome assessments and biomarkers.
And I'm Merlene Staten. I'm a long-timer of the Biomarkers Consortium, where I was co-chair of the Metabolic Steering Committee at the beginning, and where actually bone density and NIMBLE both were projects that were in the very, very early stages. My background is both. I did clinical drug development and also worked at NIGDK and NIH.
Thank you. And we do have one panelist that's joining us from across the pond. Thurston Vetter from EMA is joining us. Thurston, would you like to introduce yourself?
Yes, thank you very much. And I'm really sorry I can't be with you. Very exciting meeting, and congratulations to 20 years of achievement. So my name is Thorsten Vetter. I'm Senior Scientific Officer at the EMA in the Scientific Advice Office. And we take care, as you can imagine, about scientific advice and the qualification. And I hope we can touch on both of these platforms during our discussion. Thanks.
Great. Thank you, Thorsten. So we're going to start off with some questions up here from the panel, and then we will open it up to questions from the audience. So, Jeff, I'm going to start with you. We've heard a lot about what is the difference between reasonably likely surrogate endpoint and intermediate clinical endpoint. I think I have a better understanding of that, but can you, perhaps give us some additional clarity on that? And then the follow-up question is, from the sponsor perspective, do we have to declare it as one or another, or do we just say it's an endpoint we want to use for accelerated approval?
So let me start with the second one, and I'll come back to it also. I think it is important to specify this from the beginning because they're really fundamentally different ideas. So I think the best way to understand reasonably likely surrogate endpoints and intermediate clinical endpoints is by looking at the big picture about what's in common between the two of them. These are both endpoints that are used in the setting of accelerated approval for conditions that are serious or life-threatening, where it's not practical to measure the outcome that you would like to measure in the setting of a clinical trial, typically because the outcomes you're interested in occur a long time after a long time. So it's very difficult in the time frame of a clinical trial to measure the definitive endpoints that determine clinical benefit during the clinical trial. So in that setting, the product can qualify for accelerated approval, and then you can use a reasonably likely surrogate endpoint or an intermediate clinical endpoint. So typically, a reasonably likely surrogate endpoint would be a biomarker that is expected to predict the ultimate clinical benefit. Similarly, an intermediate clinical endpoint is a clinical endpoint that can be measured in the time frame of a clinical trial, but is measuring something that can be measured earlier than the definitive clinical endpoint that you're interested in. Or perhaps it measures something that is not as serious as the ultimate clinical endpoint, which might be, for example, survival. So in that setting, both are really used in a similar way, both used to determine efficacy that predicts the ultimate clinical benefit. An example of an intermediate clinical endpoint outside of oncology, and you've heard about these endpoints in oncology, would be for a gene therapy for a rare neurodegenerative disorder, cerebral leukodystrophy. Here, it being a gene therapy and potentially some safety issues, you want to have evidence that the treatment provides long-term benefit. And that's assessed based on normal function, survival with normal function for the long term. An intermediate clinical endpoint would be measuring that normal function survival at 2 years, and then that would be the basis for accelerated approval. Then you would conduct a confirmatory trial showing that that same normal function survival is maintained long term. So I hope that helps you understand what an intermediate clinical endpoint would be. Outside of oncology, there aren't a lot of examples of intermediate clinical endpoints, but I think there are a number of settings where that could potentially be used.
Yeah, thank you, Jeff. So, Thorsten, the next question is for you, because accelerated approval and the concept of reasonably likely surrogate endpoints is very FDA-centric. What are comparable regulatory paths and frameworks in Europe?
Thank you very much. And I think it's very much a clarification merely on terminology here, because clearly US and EU regulators have to solve the same problem, so to accelerate access to life-saving drugs. And Michelle already mentioned that in her talk. So we talk about conditional marketing authorization, and that comes with 4 conditions, basically. So a medicinal product needs to address an unmet medical need in a chronically debilitating or life-threatening condition. It must show preliminary positive benefit risk at the time of conditional marketing authorization. It must show a clear path and strategy to generate evidence for a full data set, for a full evidence set down the line with what we call specific obligations for additional studies. And the early access to the market must address a clear public health benefit. So that's kind of the framing. In terms of the endpoint that we look at, we don't have this distinction between the reasonably likely surrogate endpoint and the intermediate clinical endpoints. In terms of terminology, we tend to talk generally about intermediate endpoints. And these intermediate endpoints can be agreed and discussed in several ways. The most common way for a medicinal product specific development is to come for scientific advice. And by the way, we also have a parallel scientific advice program with FDA, which would allow medicinal product drug developers to engage with both agencies at the same time. When it comes to developing methodologies like surrogate endpoints for the common good, we clearly recommend developers using our platform, which we call qualification of novel methodologies. So here we recommend early engagement at the time when qualification plans, so the evidence generation plans to support a certain context of use for qualification, are drawn up and should be agreed with regulators. And later in the second step, these methodology developers would come to ask for a qualification opinion from the EMA. We have several examples. Maybe one example I would like to mention is that I think 2 or 3 years ago, we qualified eGFR slope as an endpoint in chronic kidney disease trials. And this has borne fruit and shows you the combination of approaches to find agreement for 2 recently approved drugs in Europe, for sparsentan and enteral budesonide, which we call Kinpeygo. So both drugs have received conditional marketing authorization first based on a reduction in proteinuria at an early time point, and then went on in the same trial to demonstrate after 2 years a relevant difference in the preservation of eGFR slope. And maybe I stop here and look forward to our discussion. Thanks.
Thank you, Thorsten. So we've heard a lot about success stories, things that are in the works. One of the stories that we have not heard yet is about the use of neurofilament light, plasma neurofilament light, as a biomarker for the approval of tofersen in ALS. So Danielle, I think I look to you can you share some learnings from that experience? And what do you think is the next step for NFL as a potential going down the surrogate road?
Sure. Sure, thanks. So just to clarify, tofersen or Qalsody is an antisense oligonucleotide targeting SOD1 mRNA. And tofersen was granted accelerated approval using neurofilament, plasma neurofilament, as a reasonably likely surrogate endpoint in SOD1 ALS. And so Jen's specific question was about the learnings from our experience using neurofilament as a reasonably likely surrogate endpoint. I'm going to describe 2. The second one is a little bit multi-pronged, so bear with me. But the first one is, unlike the example that Dennis provided with BMP, neurofilament as a response biomarker, when we started our study, there was no information, there was no data on it. We included it in our first in-human study as an exploratory biomarker, and we continued the progression of that throughout the entire clinical program. So what I'm highlighting is the importance of including exploratory biomarkers in your early trials, but the commitment to advancing those and seeing the evolution of those as they progress. So in the case of neurofilament, when we started, we actually included phosphoneurofilament heavy in our first in-human studies, and we looked at it in CSF. The reason we pivoted in our pivotal trial to plasma and neurofilament is because not biologically there was any difference, but because of the analytical performance of the tests. So when I say there is a need for inclusion of early exploratory biomarkers, it's not only about generating the biological data to support those, it's also about generating the analytical performance, the technical performance of those tests as the clinical program progresses. So that's a key learning. The second learning, and you've heard a little bit about this already from multiple speakers over the last 2 days, but the second most important learning, and I love that Jeff is sitting next to me, is early engagement with key stakeholders. And I'm going to actually describe 3 key stakeholders. One was the physician community and key opinion leaders in this space. So In the context of neurofilament and ALS, there was a significant amount of data already demonstrating that neurofilament had a strong role in the prognostic value of ALS. So the higher the levels, the more likely a patient was going to progress, or the worse survival. And we partnered with academics and physicians in the field to really build that story. What didn't exist yet was the fact that you could use neurofilament as a reasonably likely surrogate to inform on treatment response. And so that's where the partnership with CDER and CDRH, so both divisions, really enabled us to be successful. We started early. We brought them along with us in terms of the data, but also in terms of the test itself and the analytical performance of that test. And then the third key stakeholder, and I'll turn it back to you, Jen, was the testing lab. So the other key component in our success was that we partnered closely with the testing lab that we used to generate the neurofilament data as part of our trial. We did not hand off samples and let them run. We worked with them day in and day out to understand the instrument, the chain of custody. We heard about this yesterday. I think it was from Paula from the FDA. But to understand every aspect of how those samples in that pivotal trial were processed, the performance of the test, et cetera. And then, sorry, you also asked me about the future of neurofilament. Neurofilament is not specific for ALS. Neurofilament is a nonspecific biomarker of axonal damage and neurodegeneration. And neurofilament has the potential to be a very useful biomarker of axonal damage and neurodegeneration. However, for us to utilize it as such, one of the key gaps is that we've heard about this as well. There is no reference material. So every neurofilament test generates a different concentration of neurofilament, and as such, we cannot harmonize, we cannot standardize the use of neurofilament in clinical practice. So one key next step is for us to get that certified reference material. The second one is for us in the ALS world, and I hope that this will continue in other neurodegenerative diseases where it makes sense. It doesn't make sense in all neurodegenerative diseases, is to actually use neurofilament for conducting more efficient and faster trials to make early go, no-go decisions.
Great, thank you. That was a very exciting development in the field of neurodegenerative diseases. So one of the other groundbreaking advancements last year, Nicole already touched on, which is the MRD, getting unanimous approval or endorsement, I should say, to be used as a reasonably likely surrogate endpoint for multiple myeloma. So, Han, can you talk about how that's impacted the field, and what do you see as the future for MRD and MM?
Yeah, thank you. So I feel like you're very modest in stating this, but the impact on the field is really tremendous. Myeloma is a disease that is in danger of becoming victims of the success in the field. So up until the early 2010s, traditional endpoints such as disease-free or overall survival were perfectly good in any late-phase clinical trial because the prognosis for relapse refractory patients was miserable. It was probably a year. And so for bortezomib and lenalidomide and pomalidomide, those were perfectly good endpoints. But the game started to change with the introduction of immune therapies, particularly daratumumab, which is an anti-CD38 monoclonal antibody. And in the combo studies with daratumumab, we started seeing high response rates in the relapse refractory population. And in the first combo with lenalidomide study, with 4 years of follow-up, the investigational group didn't reach median progression-free survival. So suddenly, we could see the writing on the wall that we were going to get better and better at treating myeloma, but it was going to get harder and harder to approve new agents. And the two groups that did the presentations for the ODAC deserve a lot of credit because they saw the writing on the wall back then, and this technology that was probably really intended to be a monitoring test for clinical application suddenly became critically important as a tool for clinical investigation. So the endorsement and the subsequent guidance, which was just published, is going to really be important as we go for the cure. This is a disease where 20 years ago it was a death sentence, and now it's a disease. A lot of people like to use it, chronic disease phrase, but a lot of practitioners in the field have patients who have been 5-10 years off of therapy, which sounds like a cure to me, but we need to understand how to reproduce that success for the majority. And it's likely going to take many different strategies and probably a couple more new agents to come into the field. And for that to be effective, we need to have endpoints that can be applied to clinical trials before, we all grow old and retire. So this is a really important development. And I think it's going to have immediate impact on the field because readouts for the trials that are coming down the pike. A lot of sponsors anticipated this and designed MRD negativity, sustained MRD negativity as an endpoint in ongoing trials. So this is potentially going to have impact in the near term. And it's going to have, I think there's two challenges, though, we have to look ahead to. First of all, like the majority of MRD assessments that are done are not done in the context of clinical trials. They're done in clinical practice now. But there's really a very limited number of clinical decisions that are informed by MRD that are supported by prospective clinical trial data. So that's one. How are we going to incorporate that into just general clinical practice? Is it scalable to communities, to community practitioners, and so forth? And then the second is, if we are designing clinical trials for curative intent, how does MRD figure into the definition of cure? Because there's certainly a strong faction within the myeloma research community that thinks that MRD negative should be the centerpiece or the defining feature of cure. And there's another faction that feels that may be one component of a much more nuanced and complicated picture. So, but it is a tremendous advance in the field. It's going to have direct benefit for patients now, and we're really grateful for it.
Great. Thank you, Han. Michelle, Biomarker Consortium has been working on these non-invasive MASH markers. We're on track down the RLSE path. How is the qualification pathway similar or different from pursuing scientific community consensus to gain regulatory acceptance?
I'm happy to comment and would love other people's thoughts on it too. I mean, I think, it's a little bit like building the plane while you're flying it, because this is also what we're experiencing in the NIMBLE project. So, if you're going through the mounting evidence and thinking about starting at the bottom of what should be the easiest to show, and this was in the context of use of diagnostic enrichment. That was the setup of NIMBLE. And I mean, at the time when the FNIH partnership started with NIMBLE, I mean, this was very much needed. It's not that it's not needed now, but the issue is that the field has already shifted, and we're already using different non-invasive tests for diagnostic enrichment. So therefore, it makes it actually challenging to complete NIMBLE, because now the studies where you could say, okay, all comers come in, and we'll show that using these non-invasive tests compared to not using them produce a favorable outcome for diagnostic enrichment. It can be extremely challenging to do that, because now you're not really in a context where you can even ethically not offer such available tests. And so, it's a little bit of a cautionary tale to kind of get on it, I think. And I think especially hearing all these other stories, I think one of the things that's been striking to me is that, there is a narrative in the scientific community of like, oh, we'll get there, we'll complete these trials, and then we're good, like box ticked. But it takes a lot more rigor than that. So I think it is about, if we are already, you saw where we were. I mean, I explained where we are in MASH, but obviously it's not just RLSE that we're interested in, it's also the validated surrogate endpoint. So we need to already put the groundwork in now in order to get there, and hopefully in RLSE, maybe by a different pathway, or maybe by this pathway, I don't know, can get us there and ensure that we have drug development to actually build the case for VSD validated surrogate endpoint, because otherwise if we sort of delude ourselves that it will be easy and doesn't take that rigor, then we're going to be playing catch up.
So Dennis and maybe Marlene, since you were, oh yeah, Jeff, go ahead.
Yeah, I just want to add on a little bit. There's been a lot of talk at this meeting in the networking sessions about opportunities for accepting biomarkers as reasonably likely surrogate endpoints. We talk about a couple of different pathways for getting FDA acceptance of new endpoints. One would be the qualification pathway. One would be for pharmaceutical companies to submit their proposal as part of their IND. And the other is scientific community consensus. But they're not mutually exclusive, of course. And scientific community consensus and clinical community consensus is really important in acceptance of biomarkers for purposes like reasonably likely surrogate endpoints. One of the advantages of the non-invasive tests for MASH is that there's acceptance in the field that fibrosis is measured and the measurements of liver stiffness as assessing fibrosis is really important on the causal pathway of disease. If you're talking about a novel blood-based biomarker that's not as clearly associated with the causal pathway of disease, no matter how good the evidence is correlating it with outcomes, it makes it a lot harder to convince clinicians and scientists that this could be used as a reasonably likely surrogate. So I do think it's really important at the same time you acquire evidence to engage the scientific community so there's an understanding about how that biomarker relates to what's known about the pathophysiology of disease.
Thank you, Jeff. My next question is for Dennis and Marlene, if you want to jump into, since you were there in the beginnings of the DEXA project. What lessons did you learn along the way of your journey that if you could go back and do some things differently, is there anything you could have done that could have made this be a shorter process than 12 years? And what other lessons have you learned?
Yeah, if I had gone back, I probably would have decided to retire early. But no. Actually, it's been a really interesting and fun project. I think I would maybe phrase it differently and say, what advice would I give to people in the future? And in our case, I think one thing that was more difficult was to acquire the data. And as I mentioned before, I think that the fact that clinical trial data is now much more easily available and pretty much ubiquitously available for newer clinical trials. And I don't know if people have seen, but you can see at the end of any clinical trial report in a major journal, they always say that the data will be available to qualified researchers within 6 months of the publication. I think that's what's required now. And so that's one thing that will make it easier in the future. Another thing is, I think, to really think about the biomarker itself. And in our case, we were kind of blessed by the fact that bone mineral density has been around for a long time, and it hasn't really changed a lot, and that's probably not going to be the case with other biomarkers. But I think thinking a lot about how you could validate across different markers, different assays, for example, of the same compound, and then also to keep in mind that for the outcome surrogate, what FDA is interested in is the change in the surrogate and not necessarily the surrogate itself. So if you have two different assays that are calibrated differently, they might still look similar when you look at the change, percent change. So it might be make that route a little bit easier. And the other thing is I think that FDA is probably will have an easier time with the next one through, I think, because this was the first time ever, and I must say they did very well, but the guidelines for what we needed to do were fairly vague, and so I think we've defined some kind of rigor with that, so I think it'll be easier. So I think for all these reasons, I don't think it would have to take 12 years again in the future. So
That's good to hear. Merlene, you have something to add?
Yeah, I just wanted to reiterate that I think it will be easier in the future. When this project very first came up, we had to go to individual companies and say, will you share your proprietary data? And that was a huge hurdle to get them to even conceive sharing with another company, much less sharing across multiple companies. And I do think the precedent setting of how do you interact with the agency, what's necessary, is a lot clearer now. So I just wanted to reiterate that I think that's true. And the other thing I just wanted to say is, you know, you were talking about that you couldn't hold on until a patient survived, but other reasons to take a surrogate or an intermediary is things like liver biopsy. Patients don't, I mean, think about how many liver biopsies would you want to do? And the onus that was required in these trials was, I'm surprised the patients put up with it. You know, and so I think that's another real reason for needing to work on other kind of endpoints that we need.
One other thing to think about, and maybe we could ask Dr. Siegel about this, that one of our frustrations in acquiring the data was that some of the data was really hard to get because it had gone down across like 6 different companies. You know, the drug was long ago generic. I don't know whether it might be possible that there could be some mechanism whereby researchers could get the data directly from FDA for a drug like that. So maybe, Jeff, maybe you could think about that, because FDA obviously has, as does the EMA, has all the data for these studies.
I haven't heard of situations where that's been possible, but there are situations where the FDA does the analysis themselves, and then FDA can look at data from a variety of different companies. I've been involved in a project like that, and that's something that's feasible.
Yeah. That would be, yeah, might be another choice. I mean, in fact, as part of our application evaluation, the FDA did analysis of the data that we sent. They redid the analysis, so clearly that's possible to do. So that would be maybe a big lift for FDA, but that seems like a possibility.
But I guess in the MASH context, you don't have the data if the trial, you know, they didn't submit for approval because it didn't work. So there's a lot of those. So those will be missing. And unfortunately, we have very few that have actually gone. Even the one that, you know, where there's one trial that has gone to outcomes, but it was not approved because it had some safety issues. So it's very likely the FDA hadn't even received the full data set there.
I'll just comment as well, if I could. 2 things. One, I think it's important to realize as well, we've done some of these analyses as well in the Oncology Center of Excellence, but that's been a number of years ago. And, you know, given current staffing and other issues currently, I think I'm just frankly sharing. I think that sort of endeavor would be very challenging. When we have done them in the past, it's a very heavy lift. It's very data and, you know, intensive, and a lot of effort, you know, that goes into those sort of analyses. One other issue that I think that I just point out is a real challenge for a lot of future biomarkers, I think, that I've seen in the oncology space, and something actually that the myeloma space did really well, is the issue of standardization. And so oftentimes in the field, and I look at, not to shame anyone, but I look at like ctDNA development, right? And it's often done with different assays, different approaches, tumor-informed, tumor-agnostic, you know, and also, you know, different thresholds, different time assessments. And so even if, you know, we wanted to collate that data, like, there's nothing to collate. It's too heterogeneous, you know, of a data set. And so one of the things, again, that I think the myeloma field did really well was in approximately 2015-16, the International Myeloma Working Group said, this is the standard for what we're going to define MRD as. It's going to be 10 to the minus 5, and you can do this via flow, whatever. You have to meet this threshold, et cetera. And then what happened is all of the sponsors in their ongoing investigational trials for specific products used that as the benchmark. And even if it was an exploratory endpoint or a secondary endpoint, that data was still collected in a uniform, standardized way, such that then ultimately 5-10 years later, that was able to be collated and assessments, meta-analysis was able to be done in a systematic way. But one of the things I really can't underscore enough will be important going forward is the community coming together to some extent on some of these, endpoints and developing some standard metric for a way to assess it.
Yes, and Nicole, you mentioned ctDNA. And that's been another hot topic in the oncology field as being a reasonably likely surrogate or intermediate clinical endpoint. Friends of Cancer Research has been successful in bringing sponsors together who have shared their data. But clearly there are still some gaps. So what are the gaps that you think need to be filled in the ctDNA space for us to be able to use that as an endpoint for accelerated approval?
Yeah, and that's an even harder, right, topic where we're talking about different diseases, ctDNA, and different, rates of shedding, et cetera. And, different assays are being used, different time points. And so, I think it really does require coalitions and maybe disease areas coming together and say, these are the time points we're going to use, or this is a standard at which we can level set across platforms, perhaps, I think, we'll need data eventually to be able to use this. And so the sooner we can sort of, unite around some standard ways to incorporate this into our current ongoing trials, I think it would just be helpful for the field, across the board.
Great, thank you. So Jackie, for us to generate the data that we need for, to generate the weight of evidence to get regulatory acceptance, we need to do a lot of natural history studies. We need data from clinical trials. How can we better engage patients in participating in these kinds of studies?
Yeah, that's. Is that it? Okay. Patients have migrated to communities so that we can connect with each other nationwide and worldwide to social media. A lot of us live in that space now, whether it's the sepsis communities of the sepsis survivors or the osteoporosis patients. And we find a lot of support that we desperately need there. And that might be a way to, first of all, alert patients that this is even going on, is to use social media. There's even more physicians I'm seeing on social media, researchers appearing on social media. And I think that it's a real help to us patients because it makes all of this that you're doing more accessible so that we know it's going on and then we know how we can participate.
Thank you. All right, one more question for me and then we will open it up to the room. And this is basically to everyone on the panel. And that's, what do you think are the benefits or drawbacks of approaching an RSLE or biomarker qualification as part of an IND process versus doing something like in the context of a consortium?
I'll start. I think, Dennis, you gave a great example, but you also leveraged a significant amount of data that already existed. So as you think about the qualification process, there is a need to already have, and you talked about how difficult it was to get access to data. As you think about the qualification process, there is a need to have data, a large amount of data, access to the data, maybe not always a large amount of data, but there is a need to have data. I think if you balance that with the reasonably likely surrogate endpoint approach, in many cases, there isn't the same amount of data. You're pioneering when you're talking about an RLSE. And so these are distinct from the starting point based on the data that exists and how it's being utilized in drug development. So that's one perspective.
Anyone else want to comment? Thorsten, do you have anything to add from the European perspective?
Nothing much more than I already told you. Clearly there is a potential advantage in terms of the time it takes to get an agreement when you come in the EMA case for scientific advice to discuss acceptability of an intermediate endpoint for a specific development program. But clearly, we think there is a lot of synergy and added benefit in really working after the community came together on a qualification plan and working on a qualification that is really helping the whole field to develop and in the end can really help to develop a lot of innovative drugs, hopefully, and improve public health, really.
Thank you. Okay, John.
That was a terrific panel. Thanks, everyone. Terrific talks beforehand. Really appreciate it. Great way to end the symposium. I have a question for Jackie and a question for Dennis. The question for Jackie is, does the DEXA surrogate endpoint that was developed meet your needs as a patient? And if so, why? If not, is there what kind of a biomarker would you love to have for osteoporosis? That's question number one. And question number two for Dennis is, you mentioned, yay, it's a lot easier to get clinical trial data now, but you struggled through it. I'm curious how difficult it was to get through legal agreements for the data sharing. Stop laughing, Jen. And what you did to troubleshoot those legal issues, if perchance there were any.
Yes, and thank you for that question. Yes, DEXA is spot on for me as far as the surrogate endpoint, because it's, as I've had so many DEXAs, and I have another one actually coming up in a couple of weeks, to me, as a patient and not a doctor, it's just something that's very clear that can show what's going on with our bones. It's easy, it's cheap, and it's accessible for a lot of women, you know, and so, yes, it does meet the needs of being a surrogate endpoint, because, you know, waiting until there's a fracture, I mean, I can't get a new spine. I have to live with what I've got. And so being able to monitor using the DEXA is the absolute best and most right way to go. And we really do need a lot more options and drugs, and to hear that these clinical trials are going to be able to be done faster and with less people, and sort of, you know, it just opens up a whole world of possibilities for patients like me. So I hope that answered your question.
Yeah, no, and in terms of legal agreements, yeah, I mean, I'd like to say there were no problems, but the issue was quite dramatic, and I have to say that FNI, when I think back on this to maybe 2014, when we were just getting going, FNIH was really helpful to provide the resources. We as academics, if I had to work with attorneys at UCSF, you know, we'd still be writing our first data agreement 10 years later, and it still wouldn't be signed. But the FNIH, I think they were very helpful. They came up with a standard agreement. And then this is an interesting process. Steve, I don't know if you were around, Steve Hoffman. You may have been around. It may have been before you got involved. You were young, yeah. Like me, you had a lot of dark hair at that time. So did I. Anyway, but I think what they did is they had a legal agreement, and they had all the companies sign the same agreement. Which was a little bit of a challenge, of course, because you had to go to the companies. Each company had their own change, but then they were able to do that, and that did really make things much easier. And I think they really had the resources that could do something like that we couldn't have done on our own. But I really do think that since that time, because of the push for more public access to clinical trial data, you know, headed by all kinds of different groups. Jeff Drazen, when he was editor of New England Journal, for example, was very active in this field. I'm sorry, Thorsten, did you have a comment? No. Okay. Anyway, and so I think that has really changed the landscape really dramatically. And like I said, you know, in new studies, you see it all the time. But I think also companies, particularly if they have a drug that's off patent, I think would be much more likely to want to share data now. And I think it could go much more quickly, I think, the next time around. Yeah.
Thank you. We'll take another question from the audience.
Yes, there has been a lot of controversy in gene therapy field about, I had mentioned that before, about the definition of really reasonable predictive endpoint, that you discussed here. And that definition is really subjective in a sense. And there has been inconsistency in the way regulators applied what that means as a surrogate endpoint. And so I wonder if the scientific community can come together to define the definition of what is reasonable surrogate endpoint for different diseases, rather than relying on regulators to make interpretation which are subjective.
Thorsten or Jeff want to take that one?
Yeah, I can start the discussion. So if I understand right, the question is, could we have more clear definition of what would constitute a surrogate endpoint or a reasonably likely surrogate endpoint? So I'm part of a group that evaluates proposals for use of reasonably likely surrogate endpoints for accelerated approval for rare diseases. And we look at a wide variety of different conditions, neurodegenerative disorders, metabolic disorders, and so on. When the data are clear and compelling, and I would say that would be clear pathophysiologic understanding of how the biomarker fits on the causal pathway, non-clinical data that model the disease, that indicate that an effect of the drug on the biomarker predicts an effect on the clinical endpoint in that non-clinical marker, plus some clinical data showing a good correlation between the biomarker and clinical measures, constitutes a pretty strong package that we would consider to be very supportive. The places that some of the programs fall short are that sometimes the non-clinical models don't really mirror the human disorder. There are situations where the data really don't indicate how much of a change in the biomarker is necessary to predict a change in the clinical outcome. And then you're at risk of having a drug that reduces the biomarker somewhat, but not all the way to normal. And then you may approve a drug that actually doesn't provide a clinical benefit, which would be a very bad outcome. So those would be two of the big areas of uncertainty that I've seen. So it's not so a matter of a definition. I think we know what a strong package would be. It's that the data don't always measure up to what we'd like to see. But I think further definition of what's expected would be helpful.
Yeah, so I just to end, in gene therapy, we're not looking at a decrease. We're actually looking at expression of a transgene. And it's very important to define what is the percentage. How do you define, how do you define and quantitate expression of a transgene in a defined organ, et cetera. So that would be very helpful. But I think the industry can contribute to that.
Thierry?
Yeah, Thierry Sornas, AbbVie. So maybe a naive question. We have talked a lot, and I've heard a lot about the validation and the ability to define the utility and relevance to a clinical. But one thing that was just mentioned briefly is how does the reduction in patient burden and the improved access of a surrogate or a likely surrogate endpoint is factored in the qualification process or the IND review?
Can you clarify a little bit what you're asking? So yeah. How much does the access?
So how much the the reduction of the patient burden. For instance, the two examples we have heard of the neurofilament and the MASH, taking liver biopsy or lumbar puncture is a burden to the patient and reduce accessibility overall because the number of practitioners, things like that. So is it factored in the evaluation of the biomarker as a positive, as a way to supplement obviously the core validation?
So I'm not sure if I can generalize, but certainly in the situation with MASH, there's an appreciation from the clinicians at FDA and everyone at FDA that liver biopsy is an invasive procedure that in rare cases can have complications. So finding a substitute for liver biopsy that performs well is considered to be very valuable. So that would be a situation where the impact on the patient is really decisive in creating the urgency to move on a reasonably likely surrogate that would allow us to avoid liver biopsy. In terms of other situations, I'm not sure how this applies to neurofilament light. Could you maybe elaborate on that?
Yeah, it's just simply having a neurofilament measurement in plasma is much more convenient to the patient than having that in the CSF.
I think it all has to do with the performance and whether there's evidence that it reflects the way a patient functions, feels, or survives. And I think in the case of neurofilament light for SOD ALS, that the evidence was quite strong that this was a good correlate.
Brian?
Hi, Brian Baker from Regeneron Pharmaceuticals. Danielle, you had mentioned the importance of the assay that was being used to measure the biomarker. And you also mentioned that at times these biomarkers start off as exploratory biomarkers. They also may start off as pharmacodynamic biomarkers, and then they get elevated as they go through clinical development by the sponsor to potential likely surrogate endpoints. And a question that comes up in our minds often is, as they move through those stages, the level of validation that is required. Does it require the level of validation that you would put, say, a companion diagnostic through? And along the same lines, the laboratories that you run these in, for PD biomarkers, you're running them in a bioanalytical lab. Do you need to move those into a CLIA certified lab if you're going to use them as reasonably likely surrogate endpoints?
It's a great question. And I think you want to start with what is the context of use? How are you using this biomarker? For biomarkers that organizations are using in first-in-human studies to tell you about PK-PD relationships, this is primarily information that organizations use to develop models. It's not necessarily being used for future regulatory use. So someone, I forget who it was, but someone earlier today had a slide that called out clinical use, and I actually going to say that's clinical practice use, and then regulatory use. And one piece that was missing was exploratory use. So there's also exploratory use of biomarkers in early trials where the validation, and I would actually say qualification, not validation, is fit for purpose. So you don't need to go through the entire validation, analytical validation of a biomarker in your early clinical development trials, mainly because those early trials are just about the organization learning about that data, using it for potential target engagement, pharmacodynamic modeling. But as the program progresses and you now want to use that biomarker, in many cases, a target engagement biomarker and a PD biomarker are not going to be that disease-relevant biomarker. So Jeff just described that as we're thinking about reasonably likely surrogate endpoints, you want to see that link to clinical. Sometimes target engagement and pharmacodynamic biomarkers are not that. But as your program progresses and you start to demonstrate data on those disease-relevant biomarkers of treatment response, that's when the analytical validation becomes absolutely critical, and you need to find a partner that has the right regulatory understanding and use to leverage that biomarker for future engagement with the FDA. So fit for purpose is the short answer. It really does depend on the context of use. And if you're interested in utilizing it for a reasonably likely surrogate endpoint, you do need to have greater analytical validation and a strong partner that has used biomarkers in that application in the past.
Sure. And what about the environment in which they're run? Does it need to be a CLIA?
Yeah, so this is actually a question I'd love our FDA colleagues to chime in on, because it's one that we debate within our own organization and many organizations. So whether or not actually for a reasonably, you're asking, does a reasonably likely surrogate endpoint need to be run in a CLIA laboratory?
During your phase 3 clinical trial, if you're going to be submitting that evidence to support accelerated approval.
That's a great question, and I'm going to ask our FDA colleagues to comment.
Yeah, I'll start and then others can comment. But, if you are using it for endpoint, selection, we expect, two sides of that coin. One is the clinical validation, and we've talked a lot about that, but then also the analytical validation. And so we really do, if we're using it for an endpoint, really need it to be, analytically validated as well. And, there have been some challenges with that in some programs. And so it really does, it's the other side of that component. If you're using it, and that's for an endpoint, if it's earlier, if it's an exploratory endpoint, or if it's, used for, and I won't even say patient selection, patient treatment decisions, those also, really need to have confidence that the assay, the result is, appropriate to be making decisions based on. But, if you're talking about it as an exploratory endpoint, or even earlier, just, to help guide with signal finding and dose finding, that doesn't require that level of analytical rigor.
Fit for purpose in those situations, yeah, sure.
Yeah.
Yeah, I would agree with what's been said. When a reasonably likely surrogate endpoint is submitted to a drug development program as part of an IND, then typically the consult would go to the Office of Clinical Pharmacology, who would assess whether the assay is fit for purpose for that use. If it comes to the qualification program, we generally will bring in CDRH to ask them to assess the plan for analytic validation. And I can't comment specifically on when it would mandate a CLIA lab. But that would be the process we'd follow.
Okay, thank you.
Hi. So what's beautiful about this session is it's very full circle for me, because I was leading the first and only reasonably likely surrogate endpoint for kidney transplant for the FDA biomarker qualification program, which the full qualification plan is under review right now. But why it's full circle is there wasn't this precedent, and all I saw when in a consortium based was the surrogate endpoint qualification plan that Dennis and Mary co-leading, and I reached out to Dennis as we're developing ours to figure out, lessons learned. And so that was really important for how we shaped our analyses, because it was the same prospective analyses of retrospective data. And the second part, which is full circle, is Steve Piccoli taught me all the analytical information to gather from all the different centers across the globe to support that data. So because the composite biomarker we had, these are not LDTs, and it was a composite of GFR, proteinuria, biopsy, and DSA. The guidance was we just need to get the clear CAP certifications to show that they're run, and then have the 510K clearance numbers for this to support it from that analytical. But the key part with the composite was making sure that you're not overextending the fit for purpose of that assay for its intended use in the context of that composite biomarker. So expecting it to do more than its 510K clearance for. So as far as for reasonably likely, in in the context of assay that's going into that reasonably likely, you're going to want a 510K cleared at a minimum. You're not going to expect an LDT. And you're not going to want to override what its intended approval was for that assay. So, thank you. Hi, Thorsten. And we got EMA qualification opinion for it, but because they don't have reasonably likely and we didn't have the data for a fully validated, we got it qualified with EMA as a secondary endpoint prognostic for long-term graft survival. So it's a whole family here today.
All right, any other questions or comments from the audience? Any of the panelists have anything they want to add? Oh, yes, please come up to the microphone.
I'm almost embarrassed to bring this up because while all of you were being trained to save lives, I was trained to be a lawyer. But if we are looking to advance biomarkers, at least arithmetically, if not geometrically, we have to think prospectively about what the consent looks like in trials. Our colleagues in Europe have a wonderful set of regulations designed to protect participants and people. I have no problem with that. The American system is different, and I have seen whole data lakes turn into dikes by virtue of the fact that governance committees could not put their mind at ease that the consents were adequate to allow them to share the data. And so I would ask, after this tremendous conference, that we think maybe the FNIH could provide a form of language. Let me tell you, lawyers love, oh, that's the FNIH standard. Lawyers on the other side hate that because then the standard of care has been met. So that's my only remark.
Yeah, thank you. I think that is an excellent point. I know, I'm sure my other industry colleagues face the same challenges that I do, where we want to write those consents as broadly as we can for research that makes sense, that really is fit into the study and as part of the study, even if we can't say exactly what we're going to measure. So I think there are ways that we can consent participants, that we can do broader research where anonymized data can be integrated and shared. I think that's the future of biomedical research. And if we don't do that, I think we're going to be shooting ourselves in the foot. So I don't know if anybody else wants to comment. All right, well, thank you. This has been a really great session. I really appreciate all the panelists and the speakers for participating today. And I think I'm turning it over to Amanda and John.